All right, are there any questions? So last time we had started discussing microarrays, right? And what I'm going to do is, I have already sent you guys two, actually two papers, okay, and PDF files with explanations. I'm going to be discussing those in the class, right? So first I'm going to cover from these notes, okay? It won't be in that much of detail, just like the big picture, and then I will start discussing those two papers in detail, right? At least the ratio analysis paper I'll do right now, and the other one, which is about the coefficient of determination, I'll do it at the appropriate point in time, okay, after a few more lectures. But I think we are at the stage right now where we can start discussing the ratio analysis paper, right? And my goal will be, I mean, here this is like this text or these notes, it's more like the big picture, right? But your double E's here, okay, and grad students, like masters and PhD students. So I'll try to show you some of the math, right? And for that, I have the detailed explanation so that you can follow the entire paper from start to finish, right? Did anybody try to read any of those papers? Or not yet, right? I mean, you were asking me about the paper, but that's okay. That's fine. You know, you're better off not reading it in advance because I'll supply you with the additional material that you need in order to be able to understand the complete paper. So any questions so far? So I already explained to you, right, the details about how you go around, how you go about creating a microarray, right, and then use it to analyze differential gene expression between two samples, right. Now, in each and every step, there is scope for variation, right, in the microarray. Like you're doing a washing step, you're hybridizing, right, you're putting the probe on that side. The robot might put it a little bit off that point where it's supposed to go and print the probe, right? So there are a lot of there are a lot of possibilities for variations, right? So as with any random data, understanding variation is important to microarray data analysis. And a major impediment to an effective understanding of variation is the large number of sources of variance inherent in microarray measurements, right? And as you know, if you have a situation, experimental situation where there are a lot of sources of variations, right? That is a fertile ground for statisticians, right? I mean, they can do lots of work. Like, I mean, some of you are attending some of those courses, so they will come in, they'll put in all these different models, and some of them are listed here, So it says in many statistical analysis publications, the measured gene expression data are assumed to have multiple noise sources, right? Noise due to sample preparation, labeling, right? Hybridization, background fluorescence, different arrays, fluorescent dyes, and different printing locations. Okay? So that means you can do some statistical analysis by using different models for the noise, right? Gaussian noise or whatever model you want to use, right? So in the statistics literature related to bioinformatics, various approaches have been taken to quantify and treat the noise levels in a set of experiments, right? Including number one, log transform signal plus noise ANOVA models. Does anybody know what ANOVA means? It's analysis of variance, right? ANOVA. ANCOVA is analysis of covariance, and there are software packages, statistical software packages that are available for doing all this stuff. I'm not covering that because I'm not an expert in that area, and it's rather routine stuff, right? Those models are there, and then you do some analysis of variance, you get your result, right? Then there are what are called mixture models, right? Where you assume that it's a mixture of maybe two Gaussian distributions, right? The data that you're getting, right? Things like that. Then multiplicative models, ratio distribution models, rank-based models less sensitive to noise distributions, replicates using mixed models, and quantitative noise analysis. So there's a whole bunch of stuff, and probably on each one of them you'll have several papers. Because this used to be a hot topic about 10 or 15 years ago, right? The statistical analysis of microarray data, right? Towards the end of this lecture, I'll mention to you that these days we have other techniques that are used. So it's not like microarrays can still be used, but there are other ways of trying to quantify gene expression. So besides variation due to random effects such as biochemical and scanner noise, simultaneous measurement of messenger RNA expression levels via cDNA microarrays involves variation owing to system sources, including labeling bias, right? Imperfections due to spot extraction. Because remember, there is image processing involved, right? You have printed the probe at a particular location, but when you get the image, right? Maybe the robot did not put it exactly at that location, so you have to extract that information from the image, right? Then and there is also the possibility for cross hybridization, What is cross hybridization? Anybody want to take a shot at that? What is cross hybridization? You know what DNA hybridization is. Cross hybridization means even when there is not a perfect match, right? The thing might also hybridize, right? So that is going to give you information. If it's not a perfect match with the probe, still things are hybridizing. That means that's another source of variation. Okay? So even with the development of good extraction algorithms and the use of control probes at the array printing stage to aid in accounting for cross hybridization, we are left with labeling bias resulting from the fluorescent tags as systemic error. Because even the tags that you're giving, right? At the end of the day, you're basically trying to find out the red and green color, right? Differential expression. How do you know that the dyes are equally effective? The two dyes, right? That's another source of variation. So although different experimental designs target different profiling objectives, whether it be global cancer tissue profiling, like you might want to, look at how the gene expression varies, right? In normal tissue versus cancerous tissue. That would be cancer tissue profiling, right? Or a single induction experiment with one gene perturbed. Or you might want to find out that if I go and perturb one of the genes, remember this is a genetic regulatory network, right? The genes are going to interact with each other in a multivariate fashion. So if you perturb one gene, okay, what is the effect on all the other genes, right? Let's say in the disease sample versus the normal sample. So, there are a lot of possibilities, okay? So there are many different microarray experiments that can be designed. But in all of them, right, regardless of the objective, you have to do some kind of normalization to correct labeling bias, right? Because, for example, if the red dye is twice as effective as the green dye, right? So then there is a factor of 2 that you have to figure into your calculations. Otherwise, you'll get wrong results, right? So you have to do this normalization to correct labeling bias. So the objective of this first step, this normalization step, is to reduce the variation between arrays, right? And normalization is usually implemented for one individual array, right? And in that case, if it is implemented for one individual array, then it is called intra-array normalization, which is what we are going to discuss now, right? So assessment of the effectiveness of normalization has mainly been confined to the ability to detect differentially expressed genes, right? So the biologists might know that these genes should be differentially expressed, okay? So is my normalization any good? If I run the microarray and then I find these genes are differentially expressed as predicted by the biologists, right? Then probably I've done a good job, right? Or I want to do cancer classification, right? Different stages of cancer I want to classify, right? If my normalization is not good, I will probably get all garbage, right? Right? But if my normalization is good, so that would be the test of how good your normalization is, right? They're referring to it here as classification accuracy. So the simplest and the most commonly used normalization is what is called the offset method, right? So to describe it, now suppose you have a situation where you have a microarray with thousands of spots on it, or probes on it, right? But what you're doing is that the two samples that you're looking at, they are one and the same. Like you just label the same sample, okay? Take part of the same sample, label it with green, and then label it with red, right? Then if you get it to hybridize on the different spots on the microarray, right? What do you expect is going to be the ratio of the red to the green intensity, assuming everything else is working fine? What should that be? At each spot, okay? Remember, the red and green have both come from exactly the same sample, right? You've taken mRNA from let's say one sample, right? You've used it to produce complementary DNA using reverse transcriptase, like I explained last time, right? Using and incorporated the red dye. You've used this same mRNA, right? Same set of mRNAs to produce complementary DNA, but labeled with the green dye, right? So what do you expect would be the ratio of the intensities, right, of the hybridization at each spot, of red to green? Come on. Sitanshu, yeah. What do you expect? They're coming from the same sample, right? One, yeah. I mean, this is not rocket science, right? I mean, yeah, if you're taking the same sample and you just colored it differently, you assume that red over green should be 1, right? Right? So if red over green at every spot is going to be 1, right? Then if you take the log of the intensity of red over green, what should you get? 0, right? So then if you take the red over green at every spot, right? Take the log, right? and then sum it up over all the microarray spots, okay, where you have good quality hybridization. What should you get? You should get still 0, right? If you get something from 0, that means there is something wrong, right? And you have to do a correction, right? So in other words, the red versus green at every spot, if you go and plot, right, that should be a 45-degree line that goes through the origin, because red and green intensity should be the same, right? Right? If it does, so if it is not going through the origin, then you have to shift it, okay? That will be the bias that you, or the offset that you're going to have to use, right? So this is what I'm going to try and explain here using the figure and all that, okay? So to describe this offset method, let the red and green channel intensities of the kth gene be denoted by RK and GK respectively, right? So in many cases, these are background subtracted intensities, because you want to get rid of the background and see what the actual signal is, right? So in an ideal case, where two identical biological samples are labeled and co-hybridized to the array, we expect that the sum of the log transformed ratios to be 0, okay? Because the ratio at each spot should be 1, right? However, due to various reasons like dye efficiency, scanner, photomultiplier, tube control, etc., this assumption may not be true, right? And you want to correct for that. So if we assume that the two channels are equivalent, except for a signal amplification factor, then the ratio of the kth gene Tk can be calculated as follows. How? So log of Tk will be log of Rk over Gk, right? That's actually the log ratio that you're measuring at that spot. And then there is this correction term, because these guys should have all summed up to 0, right? Because you're doing it like you're basically using the same sample, right? In both red and green colors, right? So these fellows should have summed up to 0. So the second term in this equation is a constant offset that simply shifts the RK versus GK scatter plot to a 45-degree diagonal line intersecting the origin, and NQ is basically the number of probes that have measurement quality score of 1, right? That means where hybridization has taken place, right? Or good hybridization has taken place. And of course, you have to quantitatively check that, right? Now, in some case, so that's what you see in that picture, right? So if you just went ahead and plotted red and green, right, you'll see something that is offset, right? But then if you do that correction, then you'll see something that is going through the origin. Right? Again, I mean, things will be a little bit off, because there are measurement errors, other kind of errors, all right? But this offset you get rid of, you get this line, okay? Now, so in some cases, the scatter plot may not be perfectly at a 45-degree diagonal line due to the difference between the scanner's two channels. Due to the difference when the scanner's two channels may operate at different linear characteristic regions, because there might be nonlinearities, okay? So they might be operating at different regions of the operating curve, right? So you might get some more errors. So in this case, you'd probably use full linear regression instead of requiring the line to intersect at the origin may be needed, right? So in this case, you're not looking for GK equals ARK, where A is a constant, okay? You're looking for GK equals ARK plus B, or ARK minus B, right? So this is achieved by finding coefficients A and B to minimize the expectation GK minus ARK minus B square. So it's like least squares, like a least squares fit that you're trying to do. Now some microarray expression levels may have large dynamic range, right? The expression may range from like very small values to very large values, which will cause systematic scanner deviations such as nonlinear response at lower intensity range and saturation at higher intensity, right? Although data falling into these ranges are commonly discarded for further analysis, the transition range without proper handling may still cause some significant error in differential expression gene detection. And to account for this deviation, there are other variations that have been used. Instead of using linear regression, you can use locally weighted linear regression, and they call that LOS. You don't have to memorize all these. These were hot topics, as I said, 15 to 20 years ago, right? Microarrays came up out around 1995. The paper that I'm going to discuss is 1997. So during that period, I would say from 1995 to 2005, people wrote a lot of papers on microarrays, right? And you don't want to go and waste your time reading all those papers, because it's like if I know some method in statistics, I'm going to apply that to microarrays and I will write a paper, right? I'm not one of those guys because I'm not a statistician, but I'm just saying that if you're a Bayesian statistics person, you'll write something about the Bayesian approach applied to microarray data analysis, right? But I don't think there is any definitive, story on that. Okay, that this is the paper and you go and read that, right? But there are lots of papers that have been. So one of those methods is this locally weighted linear regression. Is regularly employed as a normalization method for such intensity-dependent effects. And then there is a newer method. Okay, but again, I mean, it's probably more than 10 years old, which is called LOES. It's the same algorithm except that instead of a straight line, you're going to fit a quadratic polynomial across the piecewise segments, right? And the figure shows the effect of these three normalizations, right? So the left one is without any offset, right? This is with the offset correction. This is the result of linear regression, right? Where even the line may not be 45 degrees, so you're doing this linear regression, and then this is LOES, right? Where you are actually doing an additional correction, okay? And if you want to know more about each of these methods, then you have to go and read several papers, right? They're there in the literature. If you take a look at my book, the references are listed, right? My 2007 book, I think I listed that as a reference for this course. Did I or did I not? Right? You don't. I must have, or I can give that to you. It's a 2007 book, right? It's by myself and Dr. Doherty. You can go and take a look over there. There's an extensive list of references. And go and read that. But again, if you're a master's or a PhD student, reading that is not going to help you, right? One bit, because it's like somebody is an expert in a particular area of statistics. They wrote a paper, right? You want to read stuff that is going to get you onto new problems, right? And topics for your master's or your PhD, right? You don't want to just spend 2 years of your life just reading all those papers and then find that you get nothing out of it, right? So my advice is don't go and read all those papers. We are giving you the summary here, right? Now it may be that the two channels in a CDNA microarray, so we have already discussed normalization briefly. I will discuss some more when I talk about that ratio analysis paper, right? Now, getting back to those microarray analysis, it may be that the two channels in a CDNA microarray, the red and the green channel, they represent two sources of messenger RNA, right? To be compared, or that the red channel corresponds to the source of interest while the green channel serves as the reference channel. In either case, a basic question is whether, the main question, the key question is whether the red intensity is significantly greater than, right, or less than the green intensity, right? That's how you will find out differentially expressed genes. Now such a question is naturally approached in the framework of hypothesis tests, okay? Because you will have two distributions that have some overlap, right? And you picked up a sample, you want to know whether it's coming from one distribution versus the other, okay? So for a microarray having n genes with red and green fluorescent expression values labeled by R1 through Rn and G1 through Gn respectively, the issue is whether Rk is over or underexpressed relative to Gk, right? Because you want to know the activity of the K gene. Okay, that's what you're really after, right? So now we'll get into the math, right? So let mu of Rk and sigma Rk denote the mean and standard deviation of Rk, right? Rk is the intensity of red at the kth spot, right? And similarly you define for Gk, right? So that's a random variable, right? So let mu of Gk be the mean and sigma of Gk be the standard deviation, right? And the relevant hypothesis test is the following, right? So there are two hypotheses here, the null hypothesis, and I will talk a little bit. I mean, some of you may not be familiar with hypothesis testing, so in the introductory material that I have prepared, right, I will talk about that. Okay, but let's just get through this. I will talk about that in the context of the ratio paper. So you have two hypotheses. You have the null hypothesis H0, which says that these two, the mean of red at the kth spot is the same as the mean of green at the kth spot. Okay, that's one hypothesis, which means that at the kth spot there's no differential expression, right? And the alternative hypothesis is that the two means are different. Okay? And here you can have two cases, one mean greater than the other, and vice versa, right? But let's not talk about that right now. So you want to test this hypothesis using the ratio test statistic Rk over Gk. So you will take samples of Rk and Gk, right? Based on that, you're going to conclude that either that statistically RK and GK have the same means, okay, or not, right? That's the question you have to answer. Now, there is biological plausibility for assuming that the coefficient of variation, which is the ratio of the standard deviation to the mean, right? So if you have a distribution, right, you take the ratio of the standard deviation to the mean, that is called the coefficient of variation, right? So in that paper, in that ratio analysis paper, they justify why they can make the assumption that across most of the genes, right, that is a constant, right? They make that assumption, right? So if we make the simplifying assumption of a constant coefficient of variation, meaning that at any spot, right, independent of k, right, independent of k, the ratio of the standard deviation of the red to the mean of the red at the kth spot is the constant c, right? Same thing. The ratio of the standard deviation of the green at the kth spot to the mean of the green at the kth spot is c, right? And this does not depend on k. So all the spots, across all the spots, this coefficient of variation is the same, right? Then under the null hypothesis H0, null hypothesis means red and green both have the same mean, okay? This equation implies that even the standard deviation of the red at the kth spot is going to be the same as the standard deviation of the green at the kth spot, right? And this is going to simplify things because that means you can move across all the spots, right? And you can pool data together to get whatever estimates you need. So assuming that the red intensity at the kth spot and the green intensity at the kth spot are normally distributed, right? Normally and identically distributed, then the ratio function tk at the kth spot has got this density function. Remember, because if you are doing hypothesis testing, you need to find out the probability density function, right? Then you have to see the common area, right? And figure out, if I get a new sample, does it belong to this distribution or the other one, right? So I need density function. So that's what we want to calculate, right? So we want to calculate the probability density of the ratio, and that is done in that paper, right? And I'm going to explain to you in details how you come up with this. But the important thing to notice is that because of the constant coefficient of variation assumption across all the genes, right? See that k does not appear on the right hand side, right? So you can go from one spot to the other, and you basically have the same density function for the ratio, right? That's good news because that means at each spot, right, I can think of that as a sample that is coming from the same distribution. So I will have a lot of samples over which I can, carry out the calculations because this is giving you the density function. The variable here is t, right? Because t is the ratio variable, but I need to know what the parameter c is, right, before I can write down the complete density function. So how do I get the parameter c? I have to do some kind of estimation, right? I have to take measurements of the variable t, and from that I have to get the parameter c, right? So there is some estimation theory involved. I will cover that in a few minutes, all right? But the important thing to note again is that on the right hand side this doesn't depend on k because of all the assumptions that we have been making, right, about normality and about the constant coefficient of variation. So hence this density function holds for all genes, and all ratios satisfying the null hypothesis can be pooled to estimate the parameter of the above equation. The parameter c of the above equation. And the estimate in this case is given by this quantity. Okay, you look at n different spots on the microarray, right, and look at the ratios. You carry out this computation, and this number is going to give you the value, the estimated value for C, right? And this is obtained using a maximum. And how do you do this? All right. See, the spots that you're going to use, those are what are called housekeeping genes. All right. Housekeeping genes means they are present in all cells, right? They have functions in all cells, like the ones that code for, let's say, ribosomes and things like that, right? They have the same function. So you're going to make the assumption that between the two samples, all right, the expression of the housekeeping gene will not change. Okay, that means those values should be close to 1, right? So, and obviously for the spots corresponding to those genes, the null hypothesis holds because those values have to be, the ratio has to be close to 1, right? So using that information, right, you can calculate this C hat, right, across all those spots on the microarray corresponding to the family of housekeeping genes. Right. And I will show you how you get this. How many of you are familiar with maximum likelihood estimation? Okay, some people are. Yeah, this is the maximum likelihood estimate that you get from that density function. But for the benefit of everybody else, I'm going to cover the main principle of maximum likelihood estimation and show you how you can come up with this maximum likelihood estimate. All right, any questions? So I think right now if people thought that the course was all biology, at least this is closer to your comfort zone, right? Hopefully, I mean, especially if you have taken maximum likelihood estimation, you know about hypothesis testing and so on. Okay? If you don't, again, it's no big deal. I'm going to try to explain to you in a way that you can at least understand what's going on. You won't be the expert, right, at doing this stuff, but that's going to be okay. All right, any questions? So now once you have this parameter c hat, right, this estimate c hat, you can plug it in here, and now you have the density function, right? Right? I mean, so you will have the density you'll have the density function for the ratio, right? And this holds for any of the spots. So now you can use that as a basis, right? Because really you want the ratio to be close to 1, right? You want to see whether the ratio is significantly different from 1, right? And that you can find out by basically computing the area under the curve, right? How far away you are from 1, okay, how much significance do you have? So you can work that out, right? So that's the idea. Right? Now, so in this paper, right, in this paper we are going to use the maximum likelihood estimate of C, okay? But there are other ways of getting at C, not everybody that has worked in this bioinformatics or, analysis of microarray data hasn't used the stuff from this paper, right? So they'll have other approaches, and another possible approach to arrive at C, right, would go as follows. For a microarray that is derived from two identical mRNA samples that are co-hybridized on one slide. Identical mRNA samples, that means red and green should be the expression. So at every spot on the slide, you're expecting a ratio of 1, right? So it's called a self-self experiment. The parameter C, which is the coefficient of variation of the fluorescent intensity, provides the variation of the assay. However, for any experiment to guarantee the null hypothesis condition is not always possible, and one alternative is to duplicate some or all of the clones where the same expression ratio is expected, right? And for the ratio T of expression ratios, it can be shown that sigma square of log T, right? That means the variance of the log transformed ratio, right, is given approximately equal to 4c squared, right? Again, it's shown in some publication, it's listed in our book, right? I haven't looked at that. So here, so for any given experiment with some duplicated clones, this thing is easily calculated, right? The variance of the log transformed ratio is easily calculated, and from that you can get your C. That would be another way of getting at the C, right? But for the paper that we are going to discuss, we are going to use the maximum likelihood estimate. Now, I talked about normalization also before, like how you can do that offset and all that, right? Right? Remember I mentioned at the beginning of the class? Now, for the purposes of this paper, we are going to assume that, let's say, the red channel is, let's say, more intense than the green channel or vice versa, right? In that case, how do you do the normalization, right? And this I'm going to discuss in detail. So in practical applications, a constant amplification gain may apply to one signal channel, in which case the null hypothesis will not be that red equals green. The mean of red equals m times the mean of green, okay? Because there is some scaling that has taken place, right? So under this uncalibrated signal detection setting, the ratio density has to be modified, because now you cannot be checking for values close to 1, okay? It should be values that are close to m, right? And the density will change. And if you have taken a basic course in probability and statistics, right, you know that if you multiply a random variable by a constant, right, let's say a positive constant, right, you know how the density changes. Again, if you don't know, don't worry about it, because I'll work it out here, right? But I hope you know, because they do have a 300 level course in the department where they go through all of these things, all right? And you're a double E, so you have to know some probability and statistics, all right? But I'll work it out here. I don't want you to go and waste a lot of time, making up for your earlier deficiencies, if they happen to exist. So to decide whether expression levels of a gene from two samples are significantly different, we would like to find a confidence interval. Because see, here it's not a question of checking whether the ratio is exactly 1. Okay, I want a band around 1, right? So that I can say with confidence, with a certain amount of confidence that this thing is significantly different from 1. What I measured is significantly different from 1. If it falls within that band, it is not significantly different, right? So that's what I want to really do, right? So you'd like to find a confidence interval such that within the confidence interval, the null hypothesis cannot be rejected, right? Null hypothesis is that the means are equal, right? So that is the expression ratio Tk, which is Rk over Gk of the gene under consideration is not significantly deviated from 1 if the ratio is within that confidence interval, right? And the confidence interval can be evaluated by integrating the ratio density function, right? Because you have the probability mass under the curve, you can integrate that to find out what the confidence interval is going to be. So since the confidence interval, I mean, if you're using that density function that I showed you that contains C, since the confidence interval is determined by the parameter C, one can either use the parameter derived from pre-selected housekeeping genes, right? Because for housekeeping genes, you know that the ratio is 1, right? Or you can use a set of duplicated genes if they are available in the array. Like if you take a gene for hemoglobin at location number 10, and then a gene for hemoglobin at location number 20, right? Then the ratios you get should be the same, right? If they are not, then I can do some correction for that, right? So the former confidence intervals that you do get from these housekeeping genes, they contain some levels of variation from the fluctuation of the biological system that also affect the housekeeping genes, because you're making the assumption that housekeeping genes are not going to change between the two samples, right? But maybe there are some small changes, okay? So that's going to cause some errors. While in the other case where you're using duplicated genes, obviously duplicated genes will be at different locations. Gene for hemoglobin at spot number 1 and at spot number 25, right? At different locations, you know, there is a spot to spot variation, so you will have some error because of that, right? And spot to spot variation is not avoidable if one wishes to repeat the experiment, because the next time you do it, things will change a little bit, right? Robot might put it there, but it may be a little bit off and things like that, right? The confidence interval which is derived from the duplicated genes is referred to as the confidence interval of the assay, right? And the constant CV, again CV is coefficient of variation assumption, permits all ratios satisfying the null hypothesis to be pooled together to estimate the parameter, right? That's what we did in that expression when we got the maximum likelihood estimate. However, studies indicate that even the coefficient of variation varies with the expression intensities, right? And the constant CV condition is made under the assumption then that RK, the red and green intensities, are the expression levels detected from 2 fluorescent channels, right? And the latter assumption is based on the assumption that image processing, again, I mean, there are many steps involved, right? And there are mistakes that can be made at many different steps, right? Because there is image processing involved, right? So you're making an assumption that the image processing is doing a good job, right? If that mixes things up, if that messes things up, doesn't give you the right values, again, you know, this is uncertainty or errors that are going to propagate down, right? So this assumption about good image processing is quite accurate for strong signals, right? But problems arise when the signal is weak compared to the background, right? Now, if you have weaker signals, you'll have a bigger spread, you know, because there's noise is affecting it. If it is a stronger signal, right, intensity is higher, the effect of the noise is not so pronounced, right? So you would have to account for that, right? Now, even with good image processing, the actual expression intensity, it includes the effect of the background, right? See, the intensity that you measure is going to be the signal, right, plus the background, right, minus the mean of the background. You're subtracting out the mean of the background, right? Now, the constant coefficient of variation assumption that applies to this signal here, but you're not able to measure it. It does not apply to that other signal RK that you're actually measuring, RK and GK, right? So that is going to cause problems too, right? And that's what's basically explained here, right? See, the assumption of a constant CV applies to SRK and SGK, right? Those are the actual signals, right? Not to the signals that you measure, RK and GK. And whatever analysis we did earlier is not going to be applicable, right? So the ratio analysis performed under the constant CV condition does not apply in these new circumstances. But I think the authors of that paper, they did write some other papers trying to relax some assumptions, right? Where the method can be extended to genes that are weakly expressed too. Okay, but you don't have to go and read that. When I was teaching this class in earlier semesters, there was no plant biology, so every student would have to pick up one paper and present, and some of the students pick that particular paper, right? But for this class, for this particular semester, that's not needed. We have a lot more additional material to cover, but if you're interested, you can go and take a look, right? Now, to finish up this discussion of microarrays, I also want to talk about synthetic oligonucleotide arrays. All right. See, so far we have been talking about cDNA microarrays, all right, complementary DNA microarrays. And how do you get the probes for the complementary DNA microarrays? Yeah, go ahead. No, no. For complementary DNA microarrays, how do you get the probes? No, synthetic thing is what I'm going to talk about right now. All right. Your starting point is what? Yeah, your starting point is the messenger RNA that is expressed in a tissue, right? And from that you get the complementary DNA, right? Then you get double-stranded DNA, take one strand and make it hybridize over there. So that is starting from actual biological sample, right? Now instead of doing that, DNA is a synthetic molecule, right? You can prepare it artificially in the lab. You might say that, okay, instead of working with the complementary DNA that I'm getting from messenger RNA, which might be like thousands of base pairs long, right? I'm not going to work with that. Instead, I'm going to just look at DNA sequences that are, let's say, 25 units long, right? And these are synthetically prepared ones, so they are called oligonucleotides. So at each spot, I will put a different oligonucleotide, right? Now, if you try to repeat the same kind of experiment, you have the sample messenger RNA, you got the complementary DNA, you're checking for hybridization. These are very small pieces, so one gene, right? The cDNA that you get corresponding to one of the genes is going to hybridize to many of the spots, right? And you're going to have different intensity measurements. Then what do you have to do? You have to take all that information together, right? And then combine that information, right? And make a determination about what the intensity of that particular gene is, right? And people have done that, and this is proprietary information because companies have done that, like Affymetrix and others, right? They will give you the chip, right? And they'll tell you that this is the chip, right? They have these oligonucleotides, and they have their own formula. Okay, they'll not tell you how they got to it, right? They say, okay, run this thing. This is the protocol. Do the hybridization. Measure the intensities as these spots, right? Then it's maybe intensity at spot number 1 multiplied by 2 plus intensity at spot number 7 multiplied by 3, and so it's some weighted combination that's going to give you the intensity, right? So this is a different approach using oligonucleotide arrays. So a different approach than that of immobilizing cDNA strands copied from particular mRNA transcripts onto a surface is to fabricate synthetic polynucleotides onto the surface. For the popular Affymetrix chip, again, I mean, it's probably not that popular right now, right? Maybe 10 years ago it was popular, right? Photolithography and solid phase DNA synthesis are used to synthesize desired polynucleotide probes at specified coordinates on the slide, right? Since these synthetic probes are much shorter than the genes on a cDNA microarray, they must be designed in such a way that their collective action, right? They hybridize to different locations, take all that information, put it together, provides accurate measurement of transcript targets. Here we are discussing probes consisting of only 25 bases, right? 25 mers. Somebody asked me about mers, okay? 25 monomer means one base pair, okay? Dimer is 2 and so on, trimer, tetramer, right? Pentamer and so on. So these are 25 mers, okay? And a single 25 mer may pair with a large number of messenger RNAs, right? I mean, because there might be many messenger RNAs that have that sequence in them, right? So for each mRNA of interest, there is a set of non-overlapping or almost non-overlapping 25 mer oligonucleotides to provide sequence-specific detection that can lead to accurate abundance measurement at the next stage of the process, right? Now, probes must be designed so that the final measurement for a particular gene is not unduly affected by the fact that the short probe is going to hybridize to transcripts from other genes and other kinds of RNA in the sample, right? And the basic approach is to have for each mRNA to be detected a probe set for which different probes hybridize to different regions of the mRNA, right? And in this way, the measurement for a specific mRNA is obtained via averaging across a collection of probes, thereby mitigating the effects of noise, outliers, and cross-hybridization, right? Again, this is just thinking, right? It's not rigorously done, right? But the companies, of course, they have to market their products, so they have to show that the results work out. But the stuff about how exactly they came up, about which combination of probes should be used for a particular gene, that is proprietary information. They don't release it to the general public or the end user. Now, to further mitigate the effect of cross-hybridization, two sets of probes can be used, right? You can have what are called perfect match probes, right? These are ones that are designed to perfectly match selected nucleotide sequences in the targeted mRNA. Perfectly matched means they are perfect complements of that, right? Whereas mismatched probes are derived from the perfectly matched probes by changing one base at a central location, right? So if a perfect match probe is aimed at identifying a particular messenger RNA by matching a string of bases in the messenger RNA, then it is more probable or more likely that the messenger RNA is going to hybridize to the perfect match probe rather than to the mismatch probe, because mismatch probe there is one base that has been altered, right? And it is argued that the subtraction of the intensity for the mismatch probe accomplishes the correction for both background noise and cross hybridization. Again, I mean, it is argued, but most probably if you look at the literature, these are hand waving arguments, right? It's not like proving Viterbi's algorithm or something like that, where things are rigorously done, right? So there's a lot of hand waving. So with this in mind, the expression level for a probe set on a microarray can be estimated by the average difference, where for let's say for the K gene you have the subset of all probes such that PMK, right? That means perfect match K minus mismatch K is within 3 standard deviations of the trimmed average of all differences PMJ minus MMJ, right? So you go ahead and average it out over that entire set. So PMK minus MMK values, you're averaging it out over that entire set of probes, right? And alterations of this basic average difference have also been proposed, right? And as I pointed out, a lot of this stuff is just hand waving. See, various problems have been pointed out regarding the average difference, including the fact that the mismatched probe, I mean, it shouldn't be that way, okay? But the mismatched probe signal could exceed the perfect match probe signal, right? It should not happen that way. But remember, there are a lot of sources of variations here, right? You've broken it up, broken that gene up into so many pieces, right? You have so many of these 25 MERS, all right? So you don't know for sure. Okay, now so the next topic is classification, but I will not move on to that, right? Instead, I'm going to discuss the ratio analysis paper now, right? Because I have just given you the formulas here, right? And are there any questions before I switch to the ratio analysis paper? Right, basically, in the ratio analysis paper, the focus will be on, how to come up with a probability density function. See, there are three things actually. The one thing is going to be how to do the image analysis. Okay, how do you find out the signal? How can you tell the signal from the background? Okay, number one. Then the second thing is how do you, based on measurements of the ratios at the different spots, okay, how do you come up with that parameter C, right? Once you came up with that parameter C, you will have the density function, right? So once you have the density function, then how do you come up with the interval, right? Around 1, what is the confidence interval? Such that you can say that, okay, if it falls to the right of this ratio is significantly higher than 1. If it falls to the left, significantly lower than 1, right? So there is some amount of hypothesis testing also to be done, right? So, any questions on the general discussion that we have had so far? Before I move into a discussion of those two of that ratio analysis paper and the explanations associated with it. Any questions? Okay, then I'm going to move over there. So, okay, let's start with the paper, and then I will, as I need the math, I'm going to switch to a different paper and go there. So you guys can read this stuff, all right? So in this paper, right, so the title is Ratio-Based Decisions and the Quantitative Analysis of cDNA Microarray Images, right? So this is the first paper that carried out the analysis of microarray images, right? That did image analysis on these, for these microarray images. So let's first look at the abstract to get the big picture, and then I'll start going through the paper, right? So in the abstract, they're saying that gene expression can be quantitatively analyzed by hybridizing fluoro-tagged messenger RNA to targets on a CDNA microarray. So everybody is now comfortable with that. Okay, I explained that stuff using that picture. That's what this is saying. So comparison of gene expression levels arising from co-hybridized samples is achieved by taking ratios of average expression levels for individual genes. Okay, again, nothing new here, right? Then it says a novel method of image segmentation is provided to identify the cDNA target sites, and a hypothesis test and confidence interval is developed to quantify the significance of observed differences in expression ratios, right? So is it significantly more than 1? Is it significantly less than 1? And so on. So you need a probability density function for that. In particular, the probability density of the ratio and the maximum likelihood estimator for the distributions are derived, right? And an iterative procedure for signal calibration is developed. That one is because of that one is to do some kind of normalization, which I will point out, right? So any questions about the abstract? All right, so now let's move into, so right now you have enough biology background, right? So that when you read this paper, the biology should not throw you off. Actually, nothing should throw you off because, you know, you have the biology background and you're electrical engineers, right? So the rest of the stuff is engineering, right? Especially if you have taken some of those courses on estimation and all that, you should not be thrown off at all. Okay. So now let's try to go through the paper, right? So this is the introductory section, right? It's basically elaborating on the abstract. Then section 2 is the biological background and cDNA microarray technology, right? This talks about transcription and genes and genome and so on, right? So you should be able to easily read through it. Should be no problem. If there's a problem, let me know, right? Transcription, translation, all right, and so on, okay? Then complementary DNA, hybridization, right? All of those things, right? Then you keep going. See here it says the ability to carry out studies in which the transcriptional state of a large number of genes is determined has until recently been severely inhibited by limitations on our ability to survey cells for the presence and abundance of a large number of gene transcripts in a single experiment. So the microarray basically is going to allow simultaneous measurement of the expression status of thousands of genes, right? So that has been made possible by this technology that was invented like about 20 years ago, right? Close to 20 years ago. So this biology portion, I don't think you should have any problem reading, right? And then there is this slide, right, for the microarray, right? The slide preparation. So illustration of a microarray system, all right. So you're going to have these probes on the different locations. I already explained that before, all right. Then you have from the DNA, from the messenger RNA, you're going to use reverse transcription labeling with fluorescent dyes to create like red samples, green samples, right? Then you get them to hybridize onto the microarray, right? Wash it off. Whatever has not hybridized, wash it off. And then you're going to use the green light and the red light, right, to get your image, right? And then all this part is about image processing, and then you have the image that you're now going to analyze, okay? So now you have to see at every spot, okay, there's red and green, right? So you can tell that with the naked eye. If red is significantly higher than the green, you can just look at that and say, yeah, red is higher. Green is significantly higher, you can tell. But if it is red and green are about the same, it'll probably look yellow, right? So how do you tell which one is greater, right? And that's where you need all the statistics and all that stuff, right? So next we move on to how do you extract the information that you need about the spots, all right, from the microarray. So the typical microarray image is going to look like this, right? You have like green dots, red dots, and yellow dots, right? So you are going to have, see, first of all, you know where the robot went. You may not know exactly, so based on that, you can have a target patch, okay, because you know where the robot did the spotting and all that, right? But in that target patch, all right, you are going to try to find out, so there's this target patch here, right? And there is also a target mask, right, inside which is the signal that you're interested in. Anything outside this mask is the background, right? So the key question that you have to ask, okay, before you can identify, which are the pixels that you're going to take and use for your further analysis, you're going to identify that, is the signal here significantly different from the background, right? And how do you do that? You have to compare the two, right? So you need some procedure to do that, right? And remember, the robot is moving along, it'll make some inaccuracies. Then even when you lay out the probe, right, it may not be uniform. It might, like in the circle, it might be some region here where it has, the probe has stuck, right? Maybe there is no probe here, right? No probe outside. So you have to find out the exact region, because that's what you need for figuring out the intensity when you carry out the hybridization, right? So you need to find that out, right? So how do you do that? Well, they will use, see, for segmentation and all that, they'll use standard techniques, right? But to find out where is the target site within this target mask, right? Where do you actually have the signals, right? You have to use some procedure, right? And the procedure will be as follows. You find out where is the signal over here significantly different from the background, right? That's the idea. And that is done using what is, at least in this paper, they have done this using what is called the Mann-Whitney test, right? Mann-Whitney test, or the rank sum test, right? And I'm going to explain that to you first, all right? And then I'll go back to this paper to show what's being done here, right? So let me go to my other file, the explanation file, all right? So this is the explanation file, all right? So I'm going to try to fill in the gaps, all right, as you're reading through that paper. Before that, any questions so far? So now, so we are looking at the paper Ratio-Based Decisions and the Quantitative Analysis of cDNA Microarray Images, and we're trying to fill in some of the details, all right? So the first thing I want to fill in is the Mann-Whitney test, or the rank sum test, right? So what do you do in this test, right? So here the key question that you're asking is, there are two different populations, right? And I want to know if they have similar means, okay? So what you can do is, if they have similar means, okay, you take samples from one population, right, and samples from the other, then they should be all kind of mixed up, right? So what you're going to do is, let's say you're drawing n samples from population X, right, independent samples from population X, m independent samples from Y, right? M and n don't have to be equal; they could be equal to, right? So now you're going to order these guys, all right? Like you're going to arrange them in ascending order of magnitude, okay? So there will be let's say the first one is X, right? Second one is Y, the third one is again X, okay? So the rank of the first X that you get is 1, the rank of the second X that you get is going to be 3, right? And so on, right? So you order these X's and Y's, right? And then you go and basically sum up the ranks of all the X's that you see, right? So here this should have been I equal to 1 to n actually, because I have n of the x's, all right? Right? So you go ahead and sum up the ranks of all the xi's. So order x and y in ascending order of magnitude, where rxi is the rank of, and then once you have ordered them, right? So you basically go ahead and calculate the rank. You go ahead and note the rank of each x, all right? Add them together. So that's why this is called the rank sum test, right? So this is the statistic that you're going to use, right? Now, if it so happens, right, that the x's are much smaller than y's, okay, that means the mean of x is significantly lower than the mean of y, right, really significantly lower, then when you do this arrangement, all the x's will come before all the y's, right? Agree? Right? If the population, if the mean of x is significantly less than y, then it's likely that all the x's will come before all the y's, right? So in that case, the sum that you will get will be, the sum will be 1 plus 2 plus 3 up to n, right? So this is the minimum value that w can have, right? When the mean of x is significantly lower than the mean of y, right? So that means w has got to be greater than or equal to this sum, and this is equal to n times n plus 1 over 2, right? Everybody agree? This is a formula that you should have seen in high school, right? Although once I gave this presentation, and a very senior guy told me that he didn't know this, that it was given by this formula, and he was saying no kidding and all that. So I don't know how that happened, but most of you should have seen it in high school, right? I mean, or am I wrong? Just correct me. If things have changed these days in high school, I don't know, right? Huh? It's okay means you've seen it or you have not? You have, because that's the sum of an arithmetic series, right? I mean, AP, GP, and all those things. Okay, you should have done those, right? But and I was really baffled because it was a big shot that told me that. Okay, I was giving a short course on this material. Okay, any questions so far? So now, on the other hand, the other extreme possibility is that you could have W, right? You could have a situation where the mean of the x's is significantly higher than the mean of all the y's. That means all the y's occur before all the x's, right? Right? So in that case, the rank will be, it'll start at m plus 1, right? Because all the y's will have been listed first, right? And then it's m plus 1 plus m plus 2 up to m plus n, right? And again, you can, sum this up because it's, again, the sum of the first m plus n natural numbers minus the sum of the first m natural numbers. So you get m plus n times m plus n plus 1 over 2 minus m plus 1 over 2. Agree? So w has got to be less than this number, right? These are the two extreme situations that you can have, right? So what is the mean of w? All right, we can calculate what the mean of w will be under the assumption that both the populations have the same mean, right? So let's try to calculate that. So the null hypothesis is that the two populations have the same mean, right? Mu x equals mu y, right? So under the null hypothesis, we can calculate, and I'm going to show you one of the calculations. We can calculate that the expected value of this random variable w is going to be n times n plus 1 over 2, right? And the variance of w is nm times n plus 1 over 12, right? You can do this calculation, right? And let me show you one of them, right? How do you calculate? So w is the rank sum statistic, right? It basically is the sum of the ranks, sum of the ranks of the x's, right? In that ordered sequence, arranged in ascending order of magnitude. So if you calculate the expected value of w, it will be the expected value of that summation, right? It's the expected value of that summation, right? See this summation that you have here, it's the expected value of that. But you can always bring the expectation inside the summation sign, right? Because expectation is a linear operation, right? So then, once you do that, right? So this will become sigma I equal to 1 to n, expected value of Rxi, okay? Now let's try to analyze the situation a little bit. You are saying that the x's and y's, all right, they have the same mean, right? That means it is equally likely, right, that at each location, right, each xi and each yi it's equally likely to occur in any of the positions, right? So what is the probability? So you have these m plus n items, all right? What is the probability that you will locate them in any particular position? If you pick, let's say, an xi, okay, what is the probability that the rank of that xi will be 1? Remember, they are all equally likely because the means are the same, right? The x's and y's, right? You have made the null hypothesis that the means are the same, right? So what is the probability of occupying any particular position? 1 over m plus n, right? There are m plus n numbers, all right? And each one of them is equally likely to occupy any position, right? So you have 1 over m plus n is the probability, you have that appearing in the denominator. And that's the same for each location, right? So corresponding to each rank, the probability is the same, right? So you will have, you're now taking the expectation, that means you have to multiply the rank, right, of xi with the probability of xi. So it's 1 times 1 over n plus m plus 2 times 1 over n plus n and so on up to n plus m over multiplied by 1 over n plus m. Is that clear to everybody? So now you do the calculation. Again, 1 plus 2 plus 3 plus up to n plus m. That again is the sum of the first n plus n natural numbers. So it's n plus n multiplied by n plus m plus 1 over 2. The n plus m's will cancel out, right? And you're left with n plus m plus 1 over 2, right? Notice the summation is over the i's, I going from 1 to n. So you can bring that n plus m plus 1 over 2 outside. And now you're just summing 1 n times, okay? Right? So you just get n times n plus m plus 1 over 2. Right? That is the mean. Okay. So now, I mean, statisticians have already done that, right? So you want to see is the do these populations have the same mean or not? I will calculate that value W, right? So I showed you how to calculate the mean. You can calculate the variance, all right? And then you will have a test, okay? If the W value becomes too large, right? Then they probably don't have the same mean, right? Right? I mean, you're trying to say, okay, you're trying to say the following, right? Either X and Y have the same mean, or let's say the mean of X is significantly larger than Y, okay? Now, if the value W, okay, becomes too large, that means it means that most of the X's are coming later on, right? So there would be some value. So you can actually construct a table of values W alpha, which is a function of N and M. N is the number of Xi's, M is the number of Yi's, right? Alpha is actually the confidence interval, because you don't know for sure, okay? You're going to say with this much amount of confidence, all right? Right? I can say that the means are different, right? So you can construct a table of values W, alpha, and M such that if the value that you compute, the rank sum statistic that you compute, right? And these values are available in any standard book on statistics, right? They will give you, like, for different alpha and M, you don't have to calculate, okay? Statisticians have already done that. All right, so if W happens to be, so you will just determine the level of significance, okay, with how much confidence you want to declare that. You will determine that, and that will give you the entry. So you just calculate the ranks, you will figure out the ranks, sum them together. If the W happens to be greater than this, then you will reject the null hypothesis, right, at significance level alpha, right, or with confidence 1 minus alpha, right. And in this paper they have used Nm greater than or equal to 8, because in that case this statistic, right, that you're using, right, it becomes normally distributed, right. This one is distribution free. This rank sum test is distribution free. It does not make any distributional assumptions, right. It'll just take some samples from here, some samples from here, compute the statistic, see if it exceeds that value, right, and then make a call, right. So this is what we are going to use, right. This is what we are going to use, or what is being used in that paper, right, to determine the target site, right, from the background. Okay. So first of all, any questions so far? If there are no questions, I will show you how this thing is applied in that paper to come up with the target site, right. That means the set of all those pixels that you're going to use for collecting the signal at that spot. Any questions? Okay, so let's go back to the paper. Okay, so there is quite a bit of stuff that is written here in section 3, right? But let's get to this location where it says the see, all the other stuff is standard, right? Whatever discussion is here, you can follow that. It says that the difficult image processing task, starting here, is to identify the target site, right, within the target patch, right? And I showed you in the figure, right? See figure 3 color plate. Let's look at that figure again, right? So this is the target patch, all right? You want to identify the target site inside that. This site, this is the target site. And then there's a mask over here, because the mask you're getting from, plus or minus some distance, the robot is going to stay within this region. So that you'll use to define this rough boundary that is the target mask. But then you're really interested in figuring out what are the pixels, right, from which you should take the signal for your calculations, right? So that's what we have to find out. So this target patch, right, target mask, so in this region you will pick up some spots that will give you the background, right? And I now want to pick out points in here where the mean is going to be significantly different from the mean here. Okay, that's what we want to do. So the difficult image processing task is to identify the target site within the target patch. I showed you the figure. Each target site is somewhat annular owing to how the robot finger places the cDNA on the slide and how the slide is treated. However, there is variability in this placement within the patch from image to image and from target to target, right? And this variability can be so great that the target region is simply a collection of subregions within the nominal circular target region, right? This instability in the target region is manifested in the irregular way the mRNA is hybridized to the target and the consequent irregular brightness pattern uh, created by the floors within the target site, right? So it is important that mRNA intensity be measured over these floor regions because only they correspond to probe hybridized to target areas, right? And then apparently there are difficulties with trying to use conventional methods to this problem, right? So they use the rank sum test, right? So to overcome these difficulties, the authors propose a pixel selection method which is based on the Mann-Whitney test or the rank sum test, right? And there are 3 key points associated with this approach, and I already discussed that, right? I gave you that explanation about the rank sum test, right? So, but again, it's briefly described here, right? So this is the one. So there are 2 competing hypotheses. The null hypothesis is that the mean of the x equals the mean of the y. The competing hypothesis is that the mean of x is greater than y. Okay, so you have to figure out under what circumstances you can reject the null hypothesis, right? And so the rejection of H0 is going to occur when W exceeds that critical value, which I mentioned in that explanatory notes that I provided, right? So how do you apply the method that I already outlined to this example, right? So a target site is going to be segmented from the target patch according to the following procedure, right? First, a predefined target mask is used to identify a portion of the target patch that contains the target site, right? And that you will know from the positioning of the robot and so on, right? You might be a little bit off, but you will know that, right? So outside that patch, right, that is the background, right? So the target mask is based on the geometry of the potential target area and can be constructed from specially tagged targets or other strong targets and so on. Okay, he's given some details, right? Now you randomly go and pick 8 sample pixels, right, from the known background, right? That is outside the target mask, and you call them Y1 through Y8, right? And then you select the lowest 8 samples from within the target mask and call them X1 to X8, right? And now you want to see if you, with these samples, X1 through X8, all right, in the target mask, and with these samples from the background, Y1 through Y8, can I say that the mean of these Xi's, all right, is significantly higher than the mean of those Yi's, okay? That means this is significantly different from the background, right? So you can do that using the rank sum statistic, right? The rank sum statistic is calculated for a given significance level alpha, and you can set it, okay? It's at 5% or, you know, I don't know, 0.00101%, you can set the confidence level that you want, right, or the significance level, and then you can do this test, right? So if the rank sum statistic that you compute exceeds that critical value, right, then you will say the null hypothesis has been rejected, right? Which means that the pixels that you have picked up, they have significantly higher intensity than the background, right? And you're choosing 8 samples here because, for both foreground and background, because if you use 8 samples, although this is a distribution free test, right, the distribution becomes approximately normal when m equals n greater than or equal to 8. So if the null hypothesis is not rejected, right, then what do? That means the 8 xi's that you have picked up, they're not significantly different, right? Their mean is not significantly different from those yi's, okay? So that means you have to increase the xi's, right? So throw away the lowest value among the xi's, or you know, maybe a few of the lowest values, just throw them away. Then among the remaining values, pick the lowest ones. Okay, so you've now pushed up the average a little bit more. Do the rank sum test again. If you keep doing that, okay, and the null hypothesis is never rejected, that means you have a defective location, okay, because the signal intensity is not really higher than the background, right? And you should not use that for any further processing. But if at some point in time you find out that the rank sum statistic exceeds the critical value, right, then you will basically, among the 8 xi's that you have selected, take the lowest intensity, right, as the cutoff. And then you do a thresholding. Anything above that lowest intensity, that will be in the signal area. That's how you will pick up this region. Right? So when and that's what it says here. When H0 is rejected, the target site is taken to be, so it's right here. The target site is taken to be the 8 pixels causing the rejection together with all pixels in the target mask whose values are greater than or equal to the minimum value of the 8. That's what I said right now. And the resulting site is said to be a target site of significance level alpha. And as I said before, if the null hypothesis is never rejected, then you conclude that there is no appreciable probe at the target site. And furthermore, you can also require that the Mann-Whitney target site should contain at a minimum some number of pixels for the target site to be considered valid and measured for fluor intensity. And you have two pictures over here, figure 4a and 4b, that give you examples of what kind of target sites you come up with, depending on what value of alpha you choose. All right. So if you, I think it's the previous. Okay. So if you look at, see, these are the target sites that you have selected, right? So if you are doing detection at the level alpha equal to 0.05, right? That means you're doing it with 95% confidence, all right? Then you pick up all these sites that are shown here. On the other hand, if you are more stringent, all right, then this site is not even picked up. See, it was picked up here, it's not picked up here because the signal is weak. Okay. All right. Any questions so far? Okay. So now I need to go back. I need to go back to the other explanation thing, because now we are going to calculate the probability density function of the ratio parameters, right? And I also have to do maximum likelihood estimation, right? So let me talk a little bit about hypothesis testing over there, right? I mean, that's not there covered in this paper, but let me show it to you. Right. So let's go back here, right? So now, so how many of you are familiar with something that is called the Chebyshev inequality? How many people are familiar? Okay, only 3 or 4 people, right? So I'm going to, okay, among the 3 people, are you going to be able to derive it if I ask you to do it? You cannot? Okay, then I'm going to derive it, right? I'm going to derive the Chebyshev inequality. Let me try. This is the end of the class, so even if I get stuck, you guys will help me out, okay? So I will derive, see the Chebyshev inequality, see you know that if you have the mean of a distribution, right, then if you go up to like 3 standard deviations outside, most of the probability mass is contained in there, okay? That, like for the normal distribution, you can actually calculate the probability mass, but even in a distribution-free fashion, right, you can show that if you go too far away from the mean, right, the mass is going to be pretty small, right? And that can be obtained as a consequence of the Chebyshev inequality. So let me try to derive that now. It shouldn't take me that long, but hopefully I'll be able to do it, okay? So let's just derive the Chebyshev inequality, and I'm going to write out the derivation here. All right, so suppose you have a random variable x, all right, which is greater than 0, right? Positive valued random variable x, all right? Then the expectation of x is given by, so you have a random variable x greater than 0. Let's say it has got a probability density function f x of x, all right? Then what is the expectation? So if you're given the density function, right, the random variable is positive greater than 0, so what is the expectation? So integral from 0 to infinity, right? X, f of x dx, right? I can write this integral in 2 parts, right? I can say that, okay, let epsilon be some number, right? I'm going to calculate this integral over x greater than epsilon and then x less than epsilon. So let me write down, this is the integral over x greater than epsilon, right? X f of x dx, right? Plus the integral over x less than epsilon x f of x dx, right? And I'm deleting that subscript capital X, okay, because we know what f of x is, all right? So now if you look at this expression, all right, so remember all these are positive quantities, all right? X is positive, all right? So what I'm going to do is I'm just going to say that this is greater than or equal to, all right, integral over x greater than epsilon, right, x f of x dx. Agree? I didn't lose anything. I just dropped one of these terms, all right? Now, so when you're calculating this integral, you're looking at values of x greater than epsilon, right? So that means I can actually write this further. Okay, I can write this further as this is greater than or equal to epsilon times the integral of x greater than epsilon, right? F of x dx, right? What is this quantity? If you take the probability density function, right, and you're integrating it over x greater than epsilon, what is that? It is the probability that x is greater than epsilon, right? It's nothing but the probability that x is greater than epsilon. So this is actually equal to epsilon times the probability that x is greater than epsilon, right? So now if you compare what I had at the very top, the expected value of x for a positively defined, for a random variable that takes only positive values is greater than or equal to epsilon times probability of x greater than epsilon, right? So our conclusion is that the expected value of x is greater than or equal to epsilon times the probability of x greater than epsilon, right? I didn't do anything illegal so far, all right? So now for x, I'm going to put in, suppose you have a random variable x, suppose I look at x minus mu x square, right? That's a new random variable y, okay? I'm going to apply this result to x minus mu x square. So if you look at the expected value of y, what is that? Huh? In terms of x, what is that? Mu x is the mean of x, all right? So y equals x minus mu x square. So what is the expected value of y? Variance of x, all right? So this is sigma square x, right? And this is a positively defined, this is a random variable that is only going to take positive values, all right? So now if I write down what I already derived here, this one, okay? So if I write that out, see what you get. And that's exactly the Chebyshev inequality. So you get expected value of x minus mu of x square, all right? Let's say greater than, expected value of x minus mu x square is greater than or equal to, all right? Let's say epsilon square, right, times the probability that x minus mu of x square is greater than epsilon square. Agree? All I did is instead of epsilon, I've replaced it by epsilon square. Instead of x, I have replaced it by x minus mu x square. You agree with this, right? So now this quantity here, you just now told me that that is the sigma square of x, all right? So what do you end up getting? You end up getting from here that the probability that the magnitude of x minus mu x, all right, is greater than epsilon is less than or equal to sigma square x, right, divided by epsilon square. This is the Chebyshev inequality. Okay, let's stop here for today. Do you guys recognize that this is the Chebyshev inequality? So we'll use it next time. Let's stop for today.